Hello everyone. So this is the second last session for Basics of Biology course, and in the last two sessions for this course, we will be going over all the topics that we have discussed so far as the syllabus for this subject has been completed. So what I have decided is I will divide these summary sessions into two parts. Part one is today, and part one will be following up in the next week. And in these sessions, I will just touch over the topics that were taught in these sessions previously. And I might miss out some topics, or I might have questions for which I did not discuss the parts. But in all my slides, I have put the reference to the problem solving session number where the topic was actually discussed in detail. So you can always go back to those presentations and go back to that and take your notes from this and come back and solve the questions if you find any difficulty while we are doing this session. So I'll start. So today we are going to discuss these topics. So these are actually in chronological order of how we have discussed during our sessions. So it starts from the problem solving session 1 and follows till problem solving session 5. And in my slides today I have covered these topics and we'll go over each of them one by one. And I've also put questions according to the topics. So as and when we complete a topic or a part of the syllabus, we'll have some questions which I have accumulated from different sources. All right, let's start. So the first topic or the first thing that we are going to go over is the characteristics of living organism. So we had started by looking at what was biology, and we saw that biology is a study of living organism and their vital processes, and it's mainly divided into two parts, botany and zoology. It's not how it is now. You have multiple subdivisions of each of these major divisions as well. So botany refers to the study of plants, while zoology refers to the study of animals. And these living organisms have certain characteristics which make them amenable to be called a living organism. So the characteristics are that they are made up of cells, they have complex organization, they obtain and use energy, which they use for several purposes, for example, metabolism, for growth and development, for reproduction, movement, and these living organisms always sought to evolve or adapt to their environment, and that is because they are sensitive, and as a response to their environment or a stimulus they adapt, and which leads to them evolving during the course of evolution. So all these living organisms are divided into five main kingdoms based on certain criteria, and we'll see how all the living organisms in the planet or in the nature are divided or fall in these five kingdoms of life. We have some variations which do not adapt themselves in these five kingdoms. We'll come to that later. So the first kingdom that we'll discuss is the kingdom Monera. These organisms are mainly the bacteria, and the properties of these organisms are that their cell type is prokaryotic. Their cell organization is mostly unicellular, so they are unicellular organisms which for which there is presence of cell wall. Uh, their mode of nutrition is generally absorptive, and them being phototrophic, so heterotrophic, phototrophic, or chemoautotrophic. These classification, as you can remember, they were based on what kind of source they were using as so the energy and the raw material for them to be able to produce food. And for motility, they could be either motile or non-motile. If they are motile, their so this flagellum that you see here, that is their locomotory organ. Yeah. So in kingdom Monera as well, you have three further classification. You have Archaebacteria, Eubacteria, and Cyanobacteria. Eubacteria are the bacteria that you generally study of the microorganisms, and their cellular structure is as such. So they have ribosomes and cytoplasm and a nucleoid DNA. So the main characteristic of these bacteria are that they do not have a well-defined nucleus or a nuclear membrane. The nuclear material is rather compacted into a mass called nucleoid. Other than that, they have plasma membrane, cell wall, and they also have a capsule, so an covering around the cells, which is called the capsule, and the small hair-like structure called pilus, and one longer tail-like structure called flagellum, which is used for locomotion. Other than that, the next classification is archaebacteria. These bacteria, they are found in harsh conditions, so extremely salty condition or extremely high temperature conditions. So you find archaebacteria in those regions, and it's because of their cell wall and plasma membrane that they're able to withstand such high pressures. And the last is cyanobacteria. And as you can see from the name, that they are also blue green bacteria. They're also called blue green bacteria because of the fact that they have chlorophyll, which they use for the synthesis of food. So they're also phototrophic in nature. Moving on, the next kingdom is kingdom Protista, and these are eukaryotic, and again they are mostly unicellular. For some of them, there is cell wall, while for others there is not no there is no cell wall present. These can be heterotrophic or phototrophic. So phototrophic are the ones who use light and synthesize their own food, but heterotrophism refers to the mode of nutrition where you use an already synthesized food as your raw material. You don't synthesize on your own, and that could be by ingestive methods or absorptive for phototrophic, where you absorptive for where you are engulfing the food and trying to get your nutrition completed. And again, these also could be motile or non-motile, and you can see here that it is, for example, here you have euglena with these flagella. That is what they use for motility. So kingdom Protista also have further subclassification. The first group is called protozoa. Second is called slime mold, and the third is these group of chrysophytes, dinoflagellates, and euglenoids. Protozoa are exemplified by amoeboid protozoans, who are generally found in the water bodies, and they have these pseudopodias, which they use for engulfing food. Next, you have flagellated protozoans, who have the flagella, and which because of which they can move around. And example is euglena. And these euglena or these flagellated protozoans could be free living, that is outside any host organism, or parasitic, living on an host organism. Next, you have ciliated protozoans. So instead of having one flagella, they have multiple cilia all across their body, and this you can find in parameciums, and these are generally aquatic. And the last are sporozoans, which have a spore-like state. So in their life stage cycle, they will have one stage, they will be in spore-like structure where they will be feeding on another host. For example, Plasmodium. The second classification is slime molds. So these slime molds, they have so you see they are unicellular, but they have multiple of these cells coming together in a conglomerate. So they have these aggregates, these are called plasmodium, and that's why they have many nuclei. And the last is chrysophytes or dinoflagellates or euglenoids. So these are again single-celled organisms, or they could also be multicellular, but they are generally found in freshwater sources and they are photosynthetic in nature. Moving on, the next kingdom is fungi. So again, these are eukaryotic cells, and these again could be multicellular or unicellular. They too have cell wall present, but their mode of nutrition is heterotrophic, so they feed on other materials or other food sources by absorption. They get their food, acquire their food by absorption, and these are non-motile, so they do not have any such locomotory organ. And these fungi or fungal members can be subdivided into several groups based on their mode of nutrition or based on their spore formation. So if the structure of their spore carrying sac, based on that also they can be divided into different classes, or what kind of mode of nutrition they have and how do they acquire their food. On the basis of that also they can be divided into three modes. So for example, for nutrition you have saprophytic, parasitic, or symbiotic. Symbiotic is when fungi is present along with algal component, and they both mutually benefit each other and acquire nutrition. Parasitic is when the fungi lives in a host organism and parasitizes on the host to for nutrition. And saprophytic is the normal mode when they are using other food material, generally dead material, to as their mode of nutrition. And on the basis of spore, as I mentioned, it is based on how their sac is structured. So they could be zygomycetes, they could be ascomycetes, they could be basidiomycetes or deuteromycetes. The next kingdom is Plantae kingdom, which consists of all the plants that we see around us. And plants are generally eukaryotic, and mostly they have unicellular. They have multicellular organization. They have cell wall. They are phototrophic, as we know of it, and their mode of nutrition is absorptive. So they absorb their raw materials via the roots, and these are non-motile. So, as we have just seen that. Kingdom planta are defined by these characteristics. Other than that, the most important feature for plantae kingdom is the presence of photosynthetic pigment chlorophyll, which is what is enabling them to be phototrophic in nature, which are present in plastids. And generally, they divide or they propagate via asexual mode, which is in vegetative propagation, or sexually, where they have the fusion between the gametes in the male and the female reproductive organ, that is the pollen and the ovum. The last kingdom is the Animalia kingdom, which consists of all different animals which you find in your nature, ranging from the birds to the mammals to the fishes to many spores, sponges as well. And as we know of it, that they are eukaryotic and multicellular, but the main difference between plant and animal is the lack of cell wall in animal kingdom. These are heterotrophic, and they are ingestive because they ingest their food, and they are mostly motile. So, so up till now, we had seen how the animals or how the living organisms are divided into separate classes. But among these classes, for example, Plantae or Animalia, there is further division according to their taxonomy rank. And these taxonomic rank range from kingdom, phylum, class, order, family, genus, species in the decreasing order. So you move. So what happens? So what do we see is that there are some species or some group of animals which make up species. Then there are more such species which come together that share a similar genus. They fall together in the same rank of genus. Several genus make up the family, several family make up the order, several order make up the class, and several classes make up the phylum, and which finally makes up your kingdom. So this is in decreasing order. So very common example, let's say, for example, the Indian spectacled cobra. The name of the species, the scientific name is Naja naja. So the Naja and Naja generally give you the species and genus. So the first name is the genus and the second name is the species. So that gives you this binary classification. But we can further go down the or above the family tree, and we can classify them according to the family, order, class, phylum, and the kingdom. Kingdom for here, example here will be the Animalia. And then phylum will be reptiles because snakes are reptiles and cobra is a snake. All right. Now these divisions or this ranking, they are based on certain criteria. The first being level of organization. So are they based on cells, tissues, organs, organ systems and organisms taken together? They will be classified as these different ranks. Next is the level of symmetry. So do you see a bilateral symmetry in them or do you see a radial symmetry? Symmetry means how can you divide the body of planes into different regions. For example, here in butterfly, you can see they have a bilateral symmetry. So the lateral means to have a straight line. So when you have a straight line, you have similar two similar halves. So that's a bilateral symmetry. Radial symmetry is you can divide the body plane into different regions by dividing them radially. So you have these axes on which you can divide the starfish. And then spherical, as the name says, that it would be a spherical shape, and you have these different axes to divide them into equal halves. The next is the germ layers. So germ layers being the layers of the walls. So they could be diploblastic, means having two layers, so that consists generally of endoderm and ectoderm, or triploblastic, which has a mesoderm in between these two layers. So based on what kind of germ layers you have, that also determines what kind of rank you are in. The next is the presence of notochords. Notochords, as we have seen, they are just beneath the nerve cord, and they have these neural cartilages. That presence of notochord, as we will see, also make like they make up a separate class. And the last is the segmentation. So you have segmentation of the body. So basically, how many um, like the body of the organism can it be divided into multiple parts. So that is called segmentation, and you will see them generally in annelids. So you have these several segments here, and then you have arthropoda also. You can see here along the length of their body, you can divide them into several segments. But in vertebrata, you have very minimal such region where you can divide them into different segments. Okay, so based on all these different classification layers, we can see that their animal kingdom is divided into several ranks. We so for just the first step, where we have from kingdom to phylum, we have these different criteria based on which we have these different phylums. So we have, first is, as we have seen, the level of organization. So is the animal at cellular level, or do they have proper tissue, proper organ system, or just organs? So first is cellular level, and in cellular level we have porifera or sponges. So these are sponges here. And the second being tissue or organ system and organ level. These, because they are a conglomerate of different cells, they can have symmetry where you can divide these different these body planes into different regions. And how do you do that? Based on that, you can have two divisions where you have radial and then bilateral. For the radius ones, you have coelenterata and ctenophora, while for the bilateral ones, they are further subdivided on the basis of the presence of body cavity or coelom. So if you want to see body cavity, here, this is the body cavity. So the presence of this coelom or no coelom also is a criteria. So based on the presence of or absence of coelom, you have these three classification, where the ones without the coeloms are called acoelomates or the platyhelminths. The ones which have false coeloms are called ascaris. And then you have true coeloms, based on which you have further phylums. For example, annelids, arthropods, mollusks, snails, echinodermata starfish, and hemichordata and chordata. So we'll see the last two phylum in detail. So the phylum, so hemichordata is these, which are also exemplified by banana glossus. And then you have the chordata. These are further divided into urochordata, cephalochordata, and then vertebrata. So chordata means the presence of notochords. So the hemichordate will have some form of notochord, but not exactly notochord. But phylum chordata are exemplified by the presence of chordates, among which you have these kinds, which are urochordates. These are tunicates. Then you have these cephalochordates, which have the notochords in the anterior region, and then you have the vertebrata, which are exemplified by the vertebral column. So the first two, urochordata and cephalochordata, are taken together. They are also known as the protochordata. We move to vertebrata. Vertebrata are exemplified as a phylum by the presence of vertebral column, and they can be further divided again on the basis of several features. So the first division can also, and then when we move from one phylum to the next level, we go to division. So the division is on the basis of the presence of jaw or not. So the first group is the one which lacks jaw, which is called agnatha. The second group, which bears jaw, can again be divided into further classes. So here, after division, the group that actually consists of those organisms is called a class, which is a cyclostomata. The the gnathostomata, which actually bears the jaws, the animal which bear jaw, they again can be divided again on the basis of the limbs on fins, and this division is called superclass, which is Pisces and tetrapoda. So Pisces are the fishes, and these basically bears the fins. Well, the tetrapoda, as the name suggests, they have tetrapods, so they have limbs, so four limbs at least. Or what I mean is, they have limbs, and four of them. That's why the name is tetra. In uh, Pisces or the fishes, they are again further classified on the basis of what kind of fish they are. So if they are bony fish, they're called ostichthyes. If they are cartilaginous fish, they're called chondrichthyes. And tetrapods are generally divided on the basis of their habitat, or where do they live and how do they move around. The first group here is amphibia, the ones that live on both lands and water. Reptilia, which live on land generally. Aves, which are able to fly, and mammals are the ones which are signified by bipedalism. And that's why I said at least four, because here mammals, if you go higher up in the class where you have humans, we are also mammals, but we are bipedal, means we have we have four limbs, but two of them is what we use for walking around. So this is where you can see. So you have the cyclostomata here, chondric thighs and astic thighs. Then amphibia, these are reptilia. These here is salamander or a lizard, aves and mammals. Moving on from Animalia to Plantae. Plantae, the plant kingdom is also divided into several subdivisions based on several characteristics. The first characteristics is the production of seed. So the ones that do not produce seeds, they form one class, and the one that produce seeds, they form seven other class. And the ones that do not produce seeds, they have other features. For example, the primitive one is algae, which do not have any root, shoot, leaves. If they have some root-like structures or some leaf-like structure, but not true roots, then they are mosses or bryophytes. If they have roots, stems, and leaf, but they do not produce seeds, they are called pteridophytes, and they are generally exemplified by ferns. For the ones that produce seeds, if they do not have flowering plants or they do not have flowers, they are called conifers, or they are gymnosperms, for example, conifers. And the flowering plants are called angiosperms, so they will produce seed, have true root, shoot, and leaves. But they are again divided on the basis of the cotyledons. So in their zygote or in their seed, what kind of number of cotyledons they have. If it is one, that is called monocotyledons, which is seen in rice or paddy. And if they have two cotyledons, they are called dicotyledons, which are found in tamarins. So this is broadly the classification of the living organisms, and we'll go over some questions for this topic. So the first question is, the binomial nomenclature was given by whom? Second is, in which of the following kingdom are archaea and nitrogen fixing organism classified? So we have done the first one, which is archaea, and we also know what are these organisms who are able to fix nitrogen. So based on that, you can answer this question. Next is, what is the main basis of classification in the five kingdom system? And the last is, the protists have which of these following characteristic? So these are the answers. So the first one, as you can see, the nomenclature was given, binomial nomenclature was given by Linnaeus. Archaea nitrogen fixing organisms are bacteria, so they are Monera. They belong to the Monera kingdom. The main basis of classification for the five kingdom system, which is basically the Monera, fungi, protista, animals, and plants, they are basically on the basis of what mode of nutrition they use. And then the protists, they are generally characterized by the presence of membrane-bound nucleoproteins in the cytoplasm. So they are not aggregate, free nucleic acid aggregates. They have proper cell wall and cell structure. And nucleoprotein is in direct contact with the rest of the cell membrane. No, because they also have membrane-bound nucleoprotein, and that's why we are the third one was correct. The next set of questions are: so vascular bundles are found not found in which of these? Radial symmetry is found in which of these classes or which of these group of organisms? Ascaris or esculpments are characterized by what or which of these characters? And the plant that has no seeds has seeds but no flowers or fruits. Try to remember those two classification, and then how do you define these flowers, plants with seeds further into? Okay. So the first question is vascular bundles are not found in which one? So they are bryophytes. Bryophytes being the mosses. Second is radial symmetry is found in coelent treta and ctenophora. Third is ascaris. Ascaris are the one with pseudocoelom, so they don't have true coelom, but they have metamerism in the metamers in the body. And gymnosperms are the plant which have seeds but no flowering or fruits. The ones that have flowers and fruits are called angiosperms. Okay, so now we'll move on to the next topic, which is origin of life and evolution. So we had discussed five major theories for the origin of life. The first being theory of special creation, which was something which was believed to be done by gods or religious purposes, where it was said that there was one divine being who had created the entire earth. Second was a theory of spontaneous generation or abiogenesis, where because of the presence or because of the observation of the presence of maggots on the dead meat, it was said that dead meat can also give rise to living organisms. But this theory of biogenesis or abiogenesis was disproved by these three experiments, where it was in the first experiment where it was done by Rady. You can see that you have a dead meat here, and if you leave the flask until you invite these flies stay on these dead meat, and later they reproduce and give rise to the younger ones, which are the maggots. And this is what was observed in the previous slide. But to disprove or to prove that this is not because of the dead meat, he put a cork on these on the jar, and so the flask was now sealed. Now because the flask was sealed, cork cannot, like, this is not porous, so the smell of this dead meat cannot go outside, so there was no attraction of flies. But when you put a membrane or gauze on the so when you put a gauze on this flask, because of the smell of dead meat being passed on to the environment, you have these flies which are attracted to these dead meats, and they can now give rise to faggots. So this proves that it is not the dead meat, but because of the presence, like, there has to be a fly which is to be present on the dead meat which gives rise to the maggots, and it's not the dead meat that gives rise to maggots. So this was one of the experiments that disproved the abiogenetics theory. The second was Palanzani's experiment. So here he had this broth, and he cooled the broth, and he left the flask open, and he waited, and he saw that there was growth. Now what happened here was the flask was open, so it had all these organisms being attracted to it, And because over the period of time this flask had different nutrients, so there was growth. But for the abiogenetic theory, it seemed like that if you leave the flask open, it can spontaneously give rise to living organism. So to prove this, what Spallanzani did was he heated the broth, he sealed the broth again, and as you can see, the flask being sealed had no growth until the flask was removed. And because again here you can see that it is in contact with the environment, so there was growth observed. So this also shows that there was no spontaneous generation of living organism. The final experiment that disproved the theory was the Pasteur's experiment, where again you can see that upon heating the broth, you see no bacteria to growth to happen, but you heat the broth and put it, like you tilt it. So what happens when you tilt it, you have some of this broth coming to the neck region, and this neck region are in contact with the environment, so you have some bacteria growing in the flask, which is why you see growth of bacteria in the flask or in the broth. But to show that this is because of this neck region, what he did next was he did the same experiment, but he broke off the flask neck. And because the neck was broken, here you can see that the flask is in direct contact with the environment. So you again have bacterial presence. So it was not just because of the presence of the neck, but it was because of the contact with the environment that you have the bacteria coming into being. So these theory taken together were used to disprove the abiogenesis theory. And then this gave rise to the biogenesis theory, which also says omnicellular and cellular. Basically it means that a living organism comes from a living organism, so there has to be a living organism which reproduces and gives rise to the next living organism. There were some other theories as well, which is something called cosmozoic theory, where it was believed that there were some meteoric showers from the extraterrestrial environment that had landed on the earth, and they had brought with them the required or necessary components that could give rise to the living organism. Just a second. And the theory that is mostly accepted now is the modern or chemosynthetic theory of origin of life, which is validated by several hypotheses proven scientifically, where now it is shown that the origin of life has had several events. For example, it has started with chemogenic life, so it has started with chemogeny, it moved on to biogeny, and then it moved further to cognogeny. What it means in is that life started with very simple molecules, which which then which then created led to the creation of complex organic molecules like polysaccharides, fats, polypeptides, nucleic acids. These nucleic acids, fats, polypeptide, they came together to give rise to the living organism. It started with a primitive life, and it started with the coming together of nucleic acids and nucleoproteins. The nucleic acids and proteins coming together, they form nucleic proteins. These together then form the coercivates, and this coercivate then started growing in multicellular as multicellular organism, which gave rise to the primary organism. And then it went on further to have cognogeny, which means that these primary organisms then went on to acquire the ability to have to use light and raw material to generate their own food. Then they also build their plasma membrane cell wall to become more complex to give rise to eukaryotic cell walls, and then further complex organisms. So this makes sense in the light that it started with the formation of earth till the form, and it went on to show how living organism could have come about. And then finally, what we say, as I had mentioned when I was talking about the living organism, that they have the ability to evolve. And how do that evolution happen? That also was debated. Some said that it was, for example, Lamarck. He said that it was because of the characteristics being used and disused. So the classic example of this is giraffe. So he said that there are giraffes. The giraffes have longer necks because they feed on plants, and these plants had leaves which are high above them. So they use their neck most often, and because of the neck being used most often, their necks got elongated, or the ones that had that were using their necks most frequently got their lengths elongated, and because of which they have larger length. And that's why they say that it is an acquired character, and now the organism will reproduce and then pass on these characteristics. The next was theory of natural selection, which was given by Darwin. And this said that the same principle, let's say of giraffe here, he said that there were giraffes with shorter necks, there were giraffes with longer necks. But in the given habitat where the leaves were at a higher range, only the giraffes which had longer necks could feed on those leaves, and because of which they survived and they were able to reproduce and elongate their progeny, while the ones which were with the shorter necks could not do that, and because of which these guys did not survive. So evolution had a selection for the larger neck giraffes because they were fittest in the given environment, and that's what the theory says, the survival of the fittest. And then there was mutation theory, which says that there are mutations which giving rise to these kind of variations always, because of which you have these and new variation which are suitable to the new environment as and when the environment changes. So these are the main three mechanisms of evolution which are set to drive the persistent evolution of the living organism. So again, based on this, there are some questions, and we'll try to see or answer them. So the first is an example of convergent evolution. As I have mentioned that I might not have spoken about these topics in the discussion today, but these are all discussed in the previous problem solving session, so you can always go back to them. But I have chosen some of these topics to be discussed even after these questions. So if you cannot answer it here, I'll give you a cue how to answer that in the next slide. So for example, the first question is, what is an example of convergent evolution? And these are the different examples. If you don't remember convergent evolution, I'll give you just 30 seconds, and while we are answering, we can I can give you what is convergent evolution. But I'm waiting so that you can try and remember the concept. The next is the force that initiates evolution is which of these? Third is example of an homologous organ is which of these following? And the fourth is the condition that can be explained by Lamarckism is which of these? Okay. Yeah, so I'll come to the homologous and convergent evolution in next slide. But the answers were these. So the first, for the convergent evolution, it is the wings of hawk moths and wings of hawks. Homologous organs were the arms of human and wing of a bird. The force that initiates evolution is variation, because mutation also gives rise to variation, and variation is the raw material for evolution. And the fourth question is, which of this can be explained by Lamarckism? So the first is giraffe, and I'll explain how it could be explained by Lamarckism. Second is the loss of tail from humans. So you can explain this by Lamarckism, saying that humans don't need tail, so they lost it over the period of time. Similarly, you can say that humans learned to walk on their two toes. They no longer had the requirement of the other two limbs that were not really helping them to walk. So they instead of using all the four limbs, they started using two limbs, and they started having an erected position. That's why they became bipedal. Okay, so moving on to convergent evolution and homologous. So first is convergent evolution. Convergent evolution means their function is same but the structure is different. And why does that happen? That happens because of being in the same kind of environment. So if you see the structure of this wing of a hawk moth and hawk, so it's an insect and a hawk. Hawk is a bird. So the structure of the wing for the bird and the insect, they're very different, but the function is same. They're using it both for flying. So because they need to fly, they rely on flying as a mode of locomotion. So because of their functions or their purpose being the same but the structure being different, these are convergent evolution. Is this is Not the case with teeth of domestic dog or teeth of wool, because they first of all, they belong to the same family, so their structure is very similar. Similarly, you have Geospiza fortis and these are different species of the same genus, so their wings are also similar. Moving on to homologous organs. These homologous organs, they are the same, they are structurally same, but they are functionally different. Means they are descended from the same ancestor, but they have different functions because they reside in different habitats. So here the example is human and bird. So because they are closer, you have mammals closer to apes. So you can see here by structure that they have this similar humerus, radius, ulna, and then digits. But the function differs in both. In humans, you use arms for lifting or any such purpose, but in birds they use it for, they have repurposed it as a wing and they use it for flying. Again, you can see that why wing of insect and bird is not an example of this. Again, leg of a dog and spider are very different if you see structurally, because spider is from arachnoides family or the arthropods and dogs are from mammals. So those were the main difference between convergent and homologous organ. So this was what we had mainly discussed in the problem solving session 1. There are many other topics that I haven't been able to cover in today's session, but you can always go back and refer to them. Next was cell biology. I will again reiterate that I'm just touching on the topics of these different problem solving sessions, but I'll try to cover as much as I can in this session. So cells. So we have spoken about living organism. We have classified them. We have seen how they evolve. We will now move on to how are they structurally built. So the cells is what is the structural, functional, and biological unit of a living organism. So all living being, right from a bacteria to a human, they're all made up of cells. These cells can be unicellular or multicellular. And these cells can also differentiate in the form of what kind of cell they are, prokaryotic or eukaryotic, or they could be plant cell and animal cell. But their overall structure will remain the same and the function will also remain the same. So these cells can replicate independently, and that's why they are building blocks of life. And their main need, the common structure will be they are filled with cytoplasm, they have a membrane encapsulating them, and they will be filled with, or they will have biomolecules like proteins, nucleic acids, and lipids. And these cells together, they provide structure and support to the body of the organism. And these interior, other than the cytoplasm, they can have organelles which are structured into different compartments, and we'll see how. So like I was mentioning, these are the main two characteristic of prokaryotic versus or eukaryotic cells. So prokaryotics are generally found in the bacterial, while the higher ever from the protista and fungi you will find the eukaryotic cells. And how do you differentiate between them? So you have, first of all, the main differences in eukaryotic cells, you have true nucleus. So your nucleus is surrounded by a nuclear membrane, and it also has a nucleoli. But no such well-defined structure is present in a prokaryotic cell. It has something called nucleoid. There is a mass of nuclear that the DNA is just coiled up in a mass-like structure. So that's called nucleoid. And that's why the next feature is the absence of membrane enclosed organelles in the prokaryotic cells, while you have well-defined membrane-bound organelles in eukaryotic cells. For example, Golgi complex, endoplasmic reticulum, lysosomes, mitochondria. All of these are membrane-bound organelles. Next, flagella. So they, as we had seen in during when we were doing about Monera kingdom, they are important for the prokaryotic cells, while in the eukaryotic cells, this flagella is basically for movement. So the movement becomes highly complex. You have these microtubules which allow these movements in the cells. And then prokaryotics are also characterized by the presence of glycocalyx, which is in the presence form of a capsule or a slime layer. But in eukaryotic cells, this glycocalyx is present in those cells which do not have cell wall. So cell wall is necessarily not present in eukaryotic cells. The ones that do not have cell wall, that those have glycocalyx. So that's again important here. And then plasma membrane. Again, the structural components of the plasma membrane differ between the prokaryotic and the eukaryotic cell. Other differences are in the form of ribosome, what is the constituent ribosome that are present there, what kind of level of DNA you have. The DNA in the prokaryotic cells are circular and single, while in the eukaryotic cell they are multiple and they are linear in structure. Prokaryotic cells divide by binary fission. Basically, they split up into two parts, while in the eukaryotic cells, there is mitosis that takes place that divides the cell into two equal halves. And sexual reproduction in eukaryotic cells is when they use meiosis, but in prokaryotic cells, there is so such meiosis where it divides into equal half. There's a reductional division. So your DNA contents reduces by half. There is no such things in prokaryotes. In for them, for sexual reproduction, they will have conjugation where they will directly transfer their DNA material to the other bacterial cell via a physical connection. And then other than that, eukaryotic cells, so prokaryotic cells also have these plasmids, and these plasmids, they have, for example, in bacteria, they generally have the resistance to antibiotics and such kind of cells, and these are there again. These can divide on their own. They're self-called multiplying organelles, or you can say them as a like organelle or part of the cell. And and why do these vectors or plasmids use are able to self multiply? Because of the presence of origin of replication, and we will see why, what is origin of replication if you have any confusion when we are doing about transcription and DNA replication. Okay, and the purpose I put it here is because this plasmid, which is present in the prokaryote as a extra nuclear DNA, has been repurposed by us to use in cloning. So we use this extra nuclear DNA from the bacteria for cloning the genes of our interest. So we what we do generally is, because these can self multiply, and so what we do is we remove these genes and we put our genes of interest and we put it back into the bacteria, so that when the bacteria is reproducing, this plasmid is also able to reproduce. And when that does, the gene that we want, let's say any gene of interest we want them to amplify, it can be amplified in the bacterial cells, and later we can harvest the desired protein from the bacterial cell. So I just wanted to give an idea about the recombinant DNA technology that we use. Okay, so those were about the prokaryotic and eukaryotic cells. And I had mentioned in eukaryotic cells also you have two divisions. You have animal cells and plant cells, which are divided based on several characteristics. So like shape, cell walls, presence of vacuoles, chloroplast, food storage, and the presence of centrioles. So centrioles are not present in the plant cells, but they are present in the animal cells, and they're important for the division. Food storage, animals store that as glycogen, plants store food as starch. Animal cells lack chloroplast, while plant cells are autotrophic because of the presence of chloroplast. Plant cell, as you can see here, they are characterized by the presence of these large vacuoles, which are not present in animal cell, or even if they are very small in structure. Plant cells have cell wall, which is characteristic of their structure, but it is absent in the animal cell. The animal cell, that's why, because of the because of the absence of cell wall, they do not have this fixed shape. But because plants have this rigid cell wall, they have a very fixed shape. So these are the main characteristics or divisions between the animal cells and the plant cells. Again, I have compiled some questions for these topics. So the first is, which are the following cell organelles is absent in prokaryotic cells? Next, which of these is not an example of transmembrane transport between different subcellular compartments? So here I'm trying to go over the topic of the transportation, cell transportation. And then the next is the microfilaments are composed of a protein called, so the unit constituent of microfilaments are which of these? And the next is the fluidity of plasma membrane increases with what? So plasma membranes are composed of fatty acids, and so what changes the fluidity of the plasma membrane? Fluidity, as we know that plasma membrane are these fluidic in nature, and what increases the fluidity of this plasma membrane. Okay. The first one, as I had mentioned, that prokaryotic cells lack membrane bound organelles. So all of them are absent in prokaryotic cells. Second, which said that which is not an example of transport between different subcellular compartments. So the first is trauma to thylakoid space. Second is cytoplasm to the lumen of endoplasmic reticulum. And third is mitochondrial intermembrane space and mitochondrial matrix. But the last one is, or the third one is endoplasmic reticulum to Golgi complex. So can you see the difference between these three and the third one? So all these are different spaces within the organelle. That's why they are subcellular compartments. But here, these are two organelles and the movement is across these organelles, not within these organelles. As in here, from the intermembrane space, or you have your the whatever say nutrients or ions are transported into the matrix. It is within the mitochondria. So it is within the compartment of a cell. For example, here as well in endoplasmic reticulum, from in the endoplasmic reticulum to outside cytoplasm. So across a compartment. Similarly, in chloroplast, it is between stroma to thylakoid space. But in the third one, you have endoplasmic reticulum and Golgi complex being two different organelles, and the movement is just across them. So that's why this is not a subcellular traffic or subcellular movement. And the next set, microfilaments are composed of proteins which are which had the F actin as their substitute or the structural component. And the next one is the fluidity, which increases with unsaturated fatty acids. So these are unsaturated fatty acids, and you can see here that the unsaturated fatty acids have these double bonds in between them, because of which they have low melting points. These, so a single bond is structurally strong, so they are very strong attractions, and that's why they have high melting point. But unsaturated fatty acids, because of having low melting point, they melt away faster, and because they melt away faster, it increases the fluidity of the plasma membrane. Okay. So the other set of questions are, which of these cell organelles is present in animal cells but not in the plant cells? Which of these organelle is involved in apoptosis? We know lysosomes are called suicidal bags. Basically, the cells are these organelles lead to the death of the cell. And why is the activity more prevalent here? Which of these is the reason? And the next is the oxygen and the carbon dioxide. The crossing of these gases across plasma membrane is because of which of these pathways, or because of which of this process. It should be easy because we did this in the last session as well. So the first one is connected to centrioles, and we had seen that centriole is not present in the plant cell. So centrosome is the answer, which is present in animal cell but not in the plant cell. Apoptosis is generally mediated or carried out by mitochondria. Lysosomes have hydrolytic. So centrioles are basically, you will see when we will discuss about mitosis and meiosis. So these are the ones that leads to the elongation and the movement of the DNA across the metaphase plate. So we'll come to this when we are discussing the replication or cell cycle. Yeah. So the lysosomes are called suicidal bag because of the presence of hydrolytic activity. They have hydrolases, which leads to the rupturing of the cells. And oxygen and carbon dioxide move via passive diffusion. Basically, they move via across their concentration gradient. So they move from a higher concentration to a lower concentration region. Yeah. So like I was mentioning, so we will now be discussing about cell cycle. Yeah, so cell cycle are mainly divided into these phases: interphase, G1 phase, S phase, and G2 phase. And then a major stage, which is the M phase, which is the mitotic phase, which has these further division: prophase, metaphase, anaphase, telophase. So if you have to see them, like how they actually happen during cell cycle, so you start from here, G1. In this, there is normal growth and normal metabolic growth. So what the cell is trying is to prepping it up for the replication that is to occur. Next is the DNA replication stage, where the amount of DNA is double, so that when the cell finally divides into two halves, they have equal number of DNA. So the DNA replication happens here. Then it enters G2. G2 is again further preparation for mitosis. So you will have these different cycling production and these different proteins which are helpful for moving from one stage to other. And then the cell finally enters the mitotic phase, where it undergoes all these different stages, so that the cell finally divides into two equal halves. So we'll start with the interphase. So as I mentioned, in the G1 phase, you have these cell which start to grow because you have accommodated two nucleus in it. So two compartments in it. So physically the cell starts to grow. The organelles, which have to be divided into two, the daughter cells, so they also start making copies of organelles. And so the basically all the organelles start dividing into two. Then they move to S phase. Here the cells will also have those proteins being expressed at a higher rate, which is useful for their replication stage. In replication, we will see replication also in detail later, but here the idea is to duplicate the DNA. So if your DNA is 2n in number, so it will become 4n now. And the centriole will also divide into two centriole pairs. We will see how what happens later. So the centriole will also divide. The centriole is ideally one in number, but it will now divide into two pairs, which will be helpful for the later stages, which is the mitotic phase. And then it will enter the G2 phase. Again, here the cell will continue to grow more and make more of the proteins and organelles, and it will start to reorganize its content for the mitosis. Now we in between these mitosis and all these phases, you have one phase which is called the G0 phase. This G0 phase is called the cell cycle arrest. So this happens when the cell exits G1. So before moving to S phase, cell will exit from the G1 phase, and they will remain there as non-proliferative cells. These cells will not proliferate because they are under cell cycle arrest. So these are generally the quiescent cells or the senescent cells or the cells that do not divide. These are always arrested in G0 stages. But these can also enter S phase upon Q if the cell is such that can divide upon receiving any Q. But generally, these are the fully differentiated cell, the terminally differentiated cell. They do not have to divide any further, so they enter this G0 cycle. For example, neurons. So neurons are generally in the G0 phase. Okay. But the cells, to point here, the cells that require faster growth, for example, RBC, or cells which have to be regularly remade, they don't enter G0. They move from G1 to S. If they are in G1, they will move to S phase for further division. Okay. So now we come to mitotic phase, which are divided into these four phases. We start with prophase first. So in prophase, the first thing is the chromosomes will start to condense, and as you can see, this makes them easier to pull apart. So the pulling apart of the chromosome is mediated by the centrioles. And so here first thing is the chromosome will start to condense. So you can see here the chromosome is condensing here, and you have this mitotic spindle which is forming. So this is where centriole comes into play. So you have this mitotic spindle. These are long thread-like structures which are generally formed of tubulins. So they are interconnected to each other, and these radiate from the star-like structure called centrosome. So all these single unit is, so this pair is centrosome. These individual components are called centrioles, and they give out this mitotic spindle. and the spindle will move between the centrosomes, and the centrosomes, as you can see, that they are moving towards the poles. So there were two centrioles to start with. One is moving to one side, the other one is moving to the other side. And the nucleolus, so the nuclear component, nucleoli, is disappearing. This is in the start of prophase. Then towards the later stage of the prophase, the chromosomes have become very condensed, and so they have now very compact, and the nuclear envelope has broken down. So now the DNA is in the cell, floating in the cell, and so the nuclear envelope has gone away, so it is easy for the cells to divide now. And the mitotic spindle here grows more. And now, so why it was sent, this was important, because here you have the centrosome. They capture these different chromosomes at the kinetochore on the centromere. So these, remember, these are duplicated DNA. So let's say this is one chromosome. So just to understand, there are 23 chromosomes in your in our cells. So that, let's say these are the three of those, and each of them have duplicated now, and they are connected via the kinetochore. Like they have duplicated, so they are connected, and they are connected at kinetochore, and the centrosome will connect at that kinetochore to hold them across this plate. So what will happen in the next stage, because they are being pulled, there are two kind of cores on each sister chromosomes, right? So one pole will attach to one and the other will attach to one, so that they are bound in a straight plane. This is called the metaphase plate. So in the metaphase plate is characterized by the presence of this equatorial plane, where the two kinetochores of the chromosomes are attached to microtubules on the opposite spindle poles. So once this metaphase has occurred, now you have this anaphase. In anaphase, you have these centrosomes which are connected to the kinetochores. They pull the kinetochores towards themselves. So what happens? The sister chromatids now split up because they were attached at kinetochores, and the kinetochores are now being pulled by the centrosomes. These sister chromatids separates and they are moving towards different poles, and that is happening because the protein that was holding these together have now broken, and now they will go to each poles and they will become their own chromosome, and that is what you wanted, because a mitosis is generally called a equational division. So if you started with 2n, you had now 4n. If the 4n was again pulled equally to the two planes, so they have 2n moves this side and 2n moves the other side. And when the cells, so in the last phase, which is called telophase, you have the cells forming at these two ends. Both of them will have 2n, which is equal to the one that they have started with. So the last stage is telophase, where they will start re like getting those nuclear membrane back, nucleolus membrane. All these components will start again, and then organelles will also start to distribute themselves in the two halves. So, and how will that start? It will start by the breaking of mitotic spindle first, which was holding the kinetochores there. So that starts breaking apart, and then these nuclei, that is the two nucleus, are formed because of the re-emergence of the nuclear membrane, and the nucleus and the DNA, which has started to condense, will now again started to decondense and become the string-like structure that it is normally present in. And the last step is this cytokinesis, where these two cells will split off from each other, and that is because of these presence of or something called cytokinesis, which is present by the actin or the cleavage furrow, which is mediated by actin. So you have this cleavage furrow forming between these two membranes here, and there is a contractile ring which splits off, and this one cell becomes two cells now. And for plant cells, that happens like there's a cell plate, and via that cell plate this division happens, but the idea is the same, that the cell divided to two halves. Okay. uh yeah. So the only difference here between mitotic and meiotic here is that instead of this being very simple 2 and 4n to 2n, in meiosis there is further division of these daughter cells into two daughter cells. So now you can imagine that this 4n gave rise to 2 and 2n, and then this 2n will again further divide into two halves, giving rise to n and n. So, and where do you find meiosis generally? You find it in your reproductive cells. So the sperms and the egg cells, they have n and n, because The idea is when these two gametes will fuse with each other, they will give rise to zygote with 2n. So your gametes have n haploid number of genomic content, and then the fuse, the zygote has n from the mother and n from the father, which gives rise to the 2n copies. So that's why meiosis is called reductional division, which happens by the splitting of the chromosomes into two halves, and then these sister chromatids are also separated from each other. So in meiosis, the homologous chromosomes separate and then the sister chromatids separate. In the mitosis space, it is the sister chromatid that separate from each other. Okay. So so on these topics, you can try to answer these questions. So if you want to observe chromosome structure, you can observe this best in which of these stages. Next is what is the number of DNA or DNA content in the G2 stage? G2 is after S stage. Third is which stage refers to the exit of cell from the cell cycle. And next is the stage between the meiosis 1 and meiosis 2. So meiosis 1 is meiosis 2 are the steps in the meiosis stage where you have these reductional division and then equational division. So the first, the homologous chromosome separate, that is a meiosis 1, and then the sister chromatids separate further, which is called the meiosis 2. So if you, the stage which is between these two could be called what? Yeah. So the first is metaphase, as you can correctly answered. So because you remember the chromosomes had decondensed, the condensed by this stage, and they were arranged in a plate, plane, equatorial plane, which is called the metaphase plate. So B, and because it is after S phase where the genome duplication has happened, so the DNA content is twice the number. Exiting the cell cycle is G naught. This is interkinase because interphase is the name given between to the phase which is between the cell cycle stages. So the G1 and so the G stage and the M phase. But interkinase is when you refer to the stage between meiosis 1 and meiosis 2. Okay. Next, so you observe mitosis in which of these individuals? Synapses, basically synapses means the pairing of chromosomes. So which of these chromosomes will pair with each other during the stages, cell cycles? Next is the longest stage in this cell cycle is which one? And the next is when there is an increase in the condensation of chromatin during the process of cell division, what it leads to. So euchromatin and heterochromatin are the regions which are densely packed and compactly loosely packed. So if there is condensation, which is in general the DNA is condensing, what would you observe there? Okay. So the first says mitosis, and mitosis, if the cell, if these are individuals with different level of ploidy, but the cells will undergo meiosis in all of mitosis in all of them. Synapses is the coming together of homologous chromosomes because remember, let's say in our case, if you have 23 chromosomes, you have a copy each from mother and father. So these are called homologous or chromosome because you have two sets of chromosomes for one. Like, so for example, chromosome 1 is present as two sets. So these are called homologous chromosome, and they together come, when they come together, this is called synapses. And the longest stage is interphase because if it comes to anaphase, metaphase, or any other cell cycle, it will complete the cell cycle and go away. But if it is an interphase, it is the phase where it is not undergoing division, but it will go undergo division when it receives the Q. And the last one said that what will you see when you have increased condensation? So the first thing is try to understand what is a heterochromatin and euchromatin. So heterochromatin is basically those regions which are highly condensed. And why would that be? Because these regions, they are not transcribed, so you don't have any RNA polymerase to come on them and sit. So these are transcriptionally silent, and they don't consist of more genes. As you would know that even though we have a large chunk of DNA, the genes are only very less, and they are in certain regions of the DNA. So the ones that do not contain genes, they are transcriptionally silent, and they're in very condensely packed, as opposite to euchromatin, which are very loosely packed, because these have genes which have to undergo transcription. So you have the RNA polymerase to come and sit on them. So these are loosely packed. But when the DNA as a whole is undergoing condensation to undergo replication, what happens is the difference between these condensation levels decreases, because your euchromatin is also now starting to getting condensing. So your decreased differentiation between them decreases. Okay. So up till now, whatever we had discussed, this was mainly in the problem solving session 2, as you can see here. There is more on that PPT. You can go back to that session and see for more on these topics. We now move on to the biomolecules, which was part of the problem solving session 3, and where we had discussed all of these different biomolecules, so lipid, nucleic acid, carbohydrates, and proteins in details. Today, I will just try to touch on the topics and just try to recap what these biomolecules broadly are. First of all, what are these biomolecules? They are the most essential organic molecules in living organisms, because they are responsible for maintenance and several metabolic processes. So we'll start with nucleic acid first. And nucleic acids, as you know by the name, DNA and RNA, they constitute nucleic acids. They build up the genetic material of an organism. So you have DNA or RNA, which, so for example, DNA is the majority of the genetic material for most of the organism, but some viruses have RNA as the genetic material. So this could also be a nucleic acid genetic material. Now, why do I say that? Because there is a huge debate which of these was a true genetic material, or which of these came first. Anyways, so the first nucleic acid was isolated by Frederick Mascher. And the DNA and RNA, how do you differentiate between them? First of all, DNA is double stranded helical structure, and that is double stranded because of the complementarity of the strands. So you have one strand, let's say, running in one direction, which means, let's say, it is running in 5' to 3' direction. So the other strand will be running in 3' to 5' direction, which is exactly anti-parallel to it. And you have these bases which are facing each other, and they form bonds with each other, which are hydrogen bonds, because of which they're attracted, and they are bound to each other, and as a result the DNA is double helical or double stranded in nature. Compositionally, DNA consists of a pentose sugar, which is deoxyribose, phosphoric acids, and nitrogenous bases. Ribonucleic acid, which is the RNA, on the other hand, they are single stranded, so there is no such complementary binding. And but it can sometimes show a double helical structure if the RNA folds on itself. So the same structure folds on itself, and the regions which shows complementary bind to each other, showing a double helical structure. But you always have a hairpin loop, which is the bend towards the outer side. So I'll just show it, maybe that will help. So something like this. So if you see, let's say this is the RNA. So you have this RNA winding on each top of it, and you have these regions which are forming the bonds with each other. So ultimately it will look something like this. These regions are closely bound to each other, but these have no complementary, so they will be like a bubble. So this is called hairpin-like structure. So you can see something like this. And structurally or compositionally, they will again also have phosphoric acid. The pentose sugar here is ribose, And again, it will have nitrogenous bases, and we'll see the differences in a while. So here is a basic structure of DNA. So you have, this is one strand here. So you have these deoxyribose with the phosphoric acid at the 5' end, and then nitrogenous base on the first carbon here. So this bases can be further divided into purines based on what number of components their rings have. So if they're 9-membered ring, they are purines. If they are 6-membered ring, they are pyrimidine. And then based on what kind of functional group they have, they can be further classified as adenine, guanine for the purines, and cytosine and thymine for the pyrimidine. For RNA, the difference here is in the nitrogenous bases, you will have uracil instead of thymine. Uracil is structurally different from thymine only at this position, where uracil does not have a methyl group. Thymine has a 5' methyl at the 5 position, whereas uracil doesn't have it. And on the contrary, so this is what you see in uracil, as you can see the structure here. And for DNA, as I had mentioned, the sugar is deoxyribose. So just compare this and this structure here. You will see that it is called 2' deoxyribose, means in this sugar, ribose being a 5-membered, first of all, a 5-carbon sugar, in the second prime position or the second position, there is a removal of oxygen from the carbon. So you have 2 hydrogen bound to the second carbon here, whereas in the RNA you have the ribose, and this have an oxygen in the second position as well, in addition to the third position. So that's how you differentiate between the DNA and the RNA. DNA only has hydroxyl or group at the third prime position, while ribose has 2 hydroxyl group and the second and the third site. Now RNA can be so this was RNA. This are constituent of 3 types of RNA that are found in a cell, that is transfer RNA, ribosomal RNA, and messenger RNA. Each have their own function. So transfer RNA are generally part of the translation. So this is transfer RNA, where it, as the name suggests, it transfers the amino acid which is coded by the mRNA. So mRNA being the messenger RNA. This comes from transcription of DNA. DNA has a certain constant sequence. So I'll just maybe show it here so that if you are not able to follow. So let's say your DNA has a T, G, and then something in a similar manner. So this was your DNA. This gets transcribed to give mRNA. In the mRNA, you have complementarity. By complementarity, you synthesize these strands. So this is your mRNA here. So you have so mRNA is basically transcribing this sequence. So you have ATG here as well. You will see how, like which strand is coding for this, and we'll see how. So you have this, and then this is used further for translation. And during translation, you have this ATG in the mRNA here. Let's say ATG is here. So ATG codes for methionine. So the idea is at the first position of the polypeptide chain or the protein, this methionine has to be there. And who adds the methionine to the chain of polypeptide? That is done by transfer RNA. So that's why the name of transfer RNA. So if you have to put it in a simple way, messenger RNA brings the information for the amino acid to be incorporated. Transfer RNA brings the respective amino acid onto the polypeptide chain, and ribosomal RNA makes up the ribosome where this translation actually takes place. So this is how all of these RNAs are very important, and they complement each other's function. So moving on to first, as I mentioned, the idea was that what actually serves as the genetic material was a very long running question. So one such experiment which actually tried to answer this was Griffith's experiment. So what you see here is that You have this Streptococcus pneumoniae bacteria, and they had these two kind of patterns. So you have these soft colonies or the smooth, shiny colonies, and then you have the rough colonies, which is because of the presence of a mucus coat. So there are these two kind of strain of bacteria. So what he did was he injected the living R. So living means cells which are not killed somehow. So you inject those cells into the mouse and the mouse stays healthy. And then you have this smooth shiny colonies which are called the S colonies. When you inject that into the mouse dies. So something happened that the mice was now no longer able to live. Then you kill the heat like S cells by heat, so you call them heat kill cells, and when you put them, they are equal to what you see in the R cell. The mouse are living. And the last thing what he did was that he put this heat kill cells and he put the living R cells. Now heat kill cells, mice were living. Living R cells also mice were living. So ideally mouse should have lived. Mouse should have been alive. But when you do that, you see that the mice dies. So what happened was somehow the S cells got back to their virulent stage and they could now exert their influence. So what the conclusion of the experiment was that there was something that the R strain was now transformed by the S strain, and the R strain, so R strain had this transforming principle from the heat killed S strain, because of which the R strain become virulent, and that transforming principle, whatever it was from the heat killed S to the R, is the genetic material. So there is something in the cells which can be transferred to the other, so that the property of the cell can be altered and it can behave like some other cell. But what exactly was the genetic material, it was not clear, but you could see that there was something which is able to do this exchange of the genetic component. So to identify the genetic material, this was the experiment that was carried out, which is by Avery, McLeod, and McCarthy. So what they did is, so they said, okay, then there is something in the heat kill S strain, so we'll extract all the proteins and the biomolecules and then check which of these molecules actually does the effect. So they extracted proteins, DNA, RNA, lipids, and everything else, and then they infected each of these components into the cells. And they found via this experiment that DNA was the genetic material, and this was responsible for the transformation. So they could find that DNA being the genetic material generally. And what else did they use? So to confirm, so generally in experiments what you do is, if you have a proof of concept, then it enhances your experiment. So you said that DNA is a genetic material. Now what if you also show that if you remove DNA, this does not happen? So what you do is, how do you disrupt DNA? By using DNAs. So they use several enzymes which cleave proteins, for example protease or RNAs or DNAs, and they found that when they use DNA, this transformation did not happen. But when they use RNAs and protease, this transformation could not be inhibited. So now, not only you said that DNA was a genetic material, but you also proved it by proving that when you add DNAs, it does not happen any further. So you said that, so it could, you can easily conclude here that DNA is a genetic material. But of course, as has always been the case in the history of science, if you promote or if you propose something new, you're always faced with rejection. So it was not initially accepted, but later it was concluded that DNA is the genetic material. Okay. So as I have already covered in the first few slides. So again, just going over briefly. So nucleic acids are consist of aromatic nitrogenous bases, pentose sugars, and phosphates. And these are the bases which are different between the RNA and DNA, which actually just differs at the thymine, which is uracil in the RNA. And the pentose sugars, as I've already mentioned, that they differ. They are, deoxyribose is the name because of the removal of oxygen from the second position on the deoxyribose, which is in the DNA. And okay, so this was about the nucleic acid. The next group of biomolecules which is important for living organisms are the carbohydrate. a name which I'm sure everybody would know, which is glucose, sucrose, maltose, starch, glycogen, everything else. So what are these carbohydrates? As the name suggests, structurally they are carbon, they are oxygen, and they are hydrogen. So when you see a molecule with carbon, hydrogen, and oxygen, you classify them as carbohydrates. And as I mentioned, they are found in innumerable food products. For example, fibers, starches, milks, fruits, vegetables. These can be monos. So the units of carbohydrates are called saccharides because they are generally classified as sugars. So they are called saccharides. The monosaccharide means only one sugar, so this is the basic unit of a carbohydrate. When you have two of the sugar coming together, they are called disaccharides. When you have multiple of the sugars coming together, they are called polysaccharides. And these are connected by branching, or they could be linear in nature as well. The next class of biomolecules are the lipids. So these lipids are generally of they consist of these fatty acids, and they will be amphipathic in nature generally. So they will have the lipid. So you find lipid bilayer in plasma membrane. So they are generally hydrophobic, and the hydrophobic means they are insoluble in water. So in plasma membrane, which is a region where you have these extracellular matrix and water content, in there you have a polar head, so that is interacting with the extracellular matrix or outside membrane, but the lipid is embedded inside the cell. So by like they generally are classified according to what constituent they have. So they could be just storage lipids. So you have TAG, which are called triglyceride glycerols, and these are the fats or fatty acids that you consume via food. And then you have membrane lipids. That's why I was explaining this. You have several of these lipids which are constituent of our cells. So this could be phospholipid or glycolipid. This phospho and glyco is based on what kind of additional group they have on what kind of polar group they have on their surface. So if they have a sugar, they are called glycolipids. If they have a phosphate group, they are called phospholipids. And these phospholipids, as you can see by structure, these are phosphate here and it's saccharides here for glycolipids. Further, what kind of backbone they have and what kind of additional components they have, on the basis of that also they can further divide it. For example, sphingolipids, you generally find them in brain, the sphingomyelin. Okay. So these lipids also can be again divided into two types: simple lipids and complex lipids. If you call them simple lipids, they are fats and waxes. The constituents are very simple. So here you can see, right, the fatty acids and glycerol make up this fats, and waxes are these esters of fatty acids which are with alcohol. So you have one additional alcohol group. But they become complex when you add these additional polar groups, and then again you divide them on the basis of one backbone with different polar group, with different phosphate group. So they become complex in nature. Okay. The other biomolecule that we have discussed in our sessions are the proteins. So proteins, as you can see in this structure here, you take one of these part, and if you zoom it, you find something like this. So they are all these long chains of amino acids. So these are amino acids that are connected to each other, and they then fold up in the 3D space to give rise to this kind of protein structure that you see. When you type any protein name, let's say Google, and you type structure, you see structures like this, but they unit wise they are all made up of such amino acids connected to each other by peptide bonds. And all of these amino acids further are of the same structural unit. They are basically differing from each other in this R group here. So that's what I was mentioning. So amino acids, as you can see, they are generally of this kind of structure. So this is an achiral carbon. Achiral means having four different group. Chiral group having four different groups. So you have H, R, NH2, and COOH. This end is called the C terminal. This is called the N terminal because amino group or NH2 group. H being hydrogen, and this R group changes. And because of these different R groups, you have 20 amino acids. present in the milieu of amino acids. And yeah, so we'll see all the R groups. So these R groups are as further divided into different types. So they can be polar. The type of R group gives the property to the amino acid. This R group could be sulfur containing. This R group could be polar. Then the amino acid becomes very polar in miscible in water. This could be non-polar. They could be aromatic, that gives fluorescence, that are able to emit or absorb light. They could be negatively charged. They could be positively charged. If they are positively charged, they are bound to the negatively surfaces. If they are negatively charged, they are found on the positively charged surfaces. They bind to positively charged, and these charges are also important because they are constituting, let's say, a receptor. And for receptors, changes in charges or minute changes in the electrochemical charges matter a lot. So what kind of amino acid is present at the surface determines what kind of like receptivity that receptor has. As in, if it is negatively charged, it will bind to some other component. If it is positively charged, it will bind to some other component. If it is neutrally charged, then it will bind to some other ligand. So each amino acid, just by convention, you either show it by a single letter or by the three-worded letter. For example, let's go over a few of them. Let's say glycine. So if you want to write glycine in short terms, you will have GLY or G. Similarly, alanine, you will write it as either ALA or A being the single letter. And just to go over the groups very briefly, so non-polar aliphatic. So non-polar means they are not like miscible in the water. So you have glycine with H, alanine with methyl, valine with certain modification to the methyl. So you have the trimethyl, or you can you have these component valine here. Then you have further modification to make it leucine, structural rearrangement to make it isoleucine, and then methionine with a sulfur containing group. So this was about non-polar. If you move to polar but uncharged R group, so these were aliphatic. These are polar, but the R groups are not charged. So here you have, so why they are charged? Because of the presence of this hydroxyl group or this SH group or the CO group. So these are not charged, means the electrons are not or present on one of the atom. So here you have serine, threonine, cysteine, which is again a sulfur containing amino acid, proline, which has this very rigid structure because of the cyclic nature, asparagine, and glutamine. Now you have R group which are positively charged in nature, which will have this N with a positive charge. So there is lysine, arginine, and histidine, and negatively charged R groups. So you see asparagine and glutamine, which have this CONH2. When there is rearrangement of these ions here, they lose out these NH2 groups to become COO minus, carboxyl group. So this becomes aspartate and glutamine become glutamate. So also called as aspartic acid or glutamic acid because they are negatively charged. And the other is non-polar, where again they don't dissolve in water, but the R group is aromatic in nature. That means they have these rings which are phenylalanine, tyrosine, and tryptophan. Okay. And so I had mentioned about these amino acid being bond to each other by peptide bonds. So we'll just go over how these peptide bonds are formed. So these are the peptide bond is generally a condensation reaction. What do you mean by condensation reaction? It means that you have a removal of water. How does that happen? So let's say this is one amino acid and this is the next amino acid. So the COOH group and the NH2 group face each other. And when they come closer to each other, this OH from this group and H from this group is out, moves out, and this leads to the formation of this kind of bond, CONH bond. So this is called a peptide bond. Now across this peptide bond, you have several movement which is possible. What I mean by this is, first of all, for to understand that, we need to understand that there are several carbons. So each of the carbons is given a certain name according to its location. So you have C alpha being the main carbon here. This carbon is called the C alpha. So when you say C alpha, you understand it is a central carbon of any amino acid. Now, this C alpha bond to the NH, C alpha. So remember this bond and then this bond. This bond is called a psi bond. No, sorry, a phi bond. And then a C alpha to C. So this bond and then this bond. So this bond is called a psi bond. So a C alpha C bond is called psi. A C alpha end is called a phi. And then you have this CN, this bond here. This is called omega. Now, why is this important? Because these proteins or these peptides, they are free to rotate around an axis, and this axis being 180 plus minus. So what we mean is, let's say for example, this bond can move high above the plane or down the plane. So it can move this way or it can move this way. Based on the rigidity of this movement, the protein as a whole can take a certain structure. Now, let's say for example, glycine. So glycine has, so let's say that there is a stretch of glycine. So all this glycine will have H in this part. So H here, H here, H here. H is very small, statically is very small. So all these amino acids, basically all these glycines, can move freely around its like its plane. So because of which, glycine is very easy to find in different structure. When I say different structure, I mean the secondary structure. Secondary structure means when these linear change of peptide fold upon itself. How quickly and how nicely it can fold and take different conformation is determined by the strength of these bonds. And how are the strength of bond determined? By the presence of the R group. If the R group is a bulky group, the movement of the amino acids or these bonds are very restricted. If the R group is a flexible group, these bonds can move around freely. And that's how, on the basis. And one more thing, so this individual amino acid can take up which region has or can be determined by something called Ramachandran plot, where you plot these several bonds and the movement that are allowed for each of these amino acid, and you can see how well a particular amino acid can move. And that's when you see that they are all further classified into different level. So what, when we say a linear chain of amino acid, that is called the primary or the primary protein structure. When these protein structures start folding with each other and twisting with each other, they give rise to something called secondary structure. So you have like this. So you have these structures moving around at several sites, so that give rise to alpha helix. Beta sheets are when two of these polypeptides, they are running anti-parallel to each other. Tertiary, and when you have these alpha helices and beta sheets coming together, so they form bonds with each other, they give rise to tertiary. And ultimately you have quaternary, for example hemoglobin, you have four of these tertiary structure coming together and forming a bond. And why is that important? Why do these different amino acids and proteins and peptides have to come together? Because protein has a lot of function to do individually and together. They can individually form enzymes that enhance rates reaction, because enzymes are basically so let's say there has to be a conversion from sucrose to glucose, sucrose to the monomeric unit. So if you leave it in the nature by itself, it will take some 100 years for it to convert to glucose, because the sucrose has requires that much of energy to convert itself to sucrose. But you put an enzyme to it. What enzyme does is that energy barrier which was required by sucrose to come to glucose or become glucose, that energy barrier is reduced by an enzyme. And whatever was, let's say, happening in some long years of time, it will happen very quickly, because the activation energy which is required was lessened. And that's how enzyme enhance the rate of reaction. And you can see here it can reduce as many as 10 times or 17 times. So that being enzyme, for example, here you have the substrates which come to the enzyme, which enzyme bind to enzyme and then give rise to the product. Next is it can also provide as a receptor or cell surface receptor. So here, for example, here you have these proteins present on the membrane is not just the glycolipids, but they are have several proteins which are present transmembrane, like between the membrane, on the extracellular, facing the extracellular part, facing the intracellular part, and then binding to different kinds of the ligands. And then you also have proteins which are uh, which characteristically bind reversibly to a protein. For example, hemoglobin. Hemoglobin task is to carry oxygen from where to where, from lungs to the cells. So if it doesn't bind oxygen reversibly, then it will not be able to release oxygen at the site of the cells. So it binds to oxygen, yes, but it binds to itself reversibly. But you also have some protein which do not bind reversibly. They bind, and the only way that these proteins and the receptor ligands can be separated from each other are by the disruption of the component. And then lastly, you also have phagocytosis. For example, here you have in the uh, for example here, so you have phagocytosis where you have this engulfment of food particles. So you will have several proteins here which will bind to this food, like bind to each other and engulf the food material which is to be taken. Or you can have, this is for food material, you can have several components, for example, that things which are destined for apoptosis. So for those, they have to be bound to the such kind of vacuoles or such kind of pits, and then you have these pits bringing these into the lysosomes to further initiate the disruption. So all these and several other functions of proteins were discussed in the problem solving session 4, and you can go back and refer to those slides. Okay, so on the basis of the lipids and the protein discussion that we have had some questions here. So the first is glycolipids in the plasma membrane are located. So glycolipids, remember, these glycolipids are polar in nature. So try to understand where could they be present in the plasma membrane. Next is most abundant RNA in the cell. Which of these could be the most abundant RNA? Next is which of these is a phospholipid uh, type of phospholipid? And the next is which is the simplest amino acid? So we had discussed this at least for the first and the last amino acid, we had gone over it. And simplest will be the one which has the least hindrance or the least complex R group. Okay. Yeah, so the first is glycolipid. It is to be present in the outer leaflet of the plasma membrane because it has to face the extracellular matrix so that it can interact with the proteins there. Next is which of these is the most abundant RNA? rRNA, because rRNA is a constituent of ribosomes. mRNA is transcribed by DNA, and tRNA are only 20. But rRNA is a ribosomal rRNA which is present in abundance. Phospholipid is lecithin because sterol or cholesterol or less steroid, they're all steroids, they're all lipids. But phospholipids means the one, so lecithin has a phosphate group attached to it. And the simplest amino acid is glycine, which has only H as its R group. Next, again on the same topics, which of these is correct with referred to enzymes? So you the enzyme is made up of certain constituents. So which of these is correct? Here we have several options for Ramachandran plot. So based on the discussion, the brief discussion that I we just had, what could be the possible answer? Again, if talking about enzyme, which of these is incorrect? And then the last is which of these is least likely to involve in the stabilizing of a 3D folding? So when you have multiple alpha helix and beta sheets coming together, which of these could be potentially like com- con- like contributing the least to their stabilization? Yeah. So the first one is enzymes. When the enzymes work, they are called the holoenzyme. And this is something that we would have covered when we were talking about DNA polymerase, where you required the core group, apoenzyme plus the coenzyme cofactors. Next is for Ramachandran plot, you cannot deduce a sequence. We had discussed this in that problem solving session also. This is because of Edmond degradation. There's a procedure. And in Ramachandran plot, it says that it is not possible to conclude whether a peptide adopts a helical or beta sheet conformation. It is wrong, because that's the purpose of Ramachandran plot, to see in which secondary structure the amino acid is most likely to be in. So that's wrong because it says it's not possible. Third, it says the peptide that are unstructured will have all the backbone dihydrogens again in the discelled out region, which is also not correct, because it says it's unstructured, but what we mean rather is, if the of the each individual amino acid, how do that take the different conformation. The correct answer is that we can deduce the occurrence, so how frequently or how probable the amino acid is to be found in a beta turn conformation or the beta sheet conformation. Similarly, for enzymes, they are highly specific. They have to be, because they interact with specific substrate. They are denatured at higher temperature, yes, because they are made of proteins, so they get denatured if you handle them uh, at loose. They of course require optimum pH and temperature, because again, for being specific, they have to have specific conditions requirement. But enzymes are proteins. They can also be RNA, but they are not lipids in nature. The enzymes which are of RNA in nature are called ribozymes. And the last one is, which of these interactions are the least contributing? So you have electrostatic hydrogen and ester. These are stronger bonds as compared to hydrophobic. So you have hydrophobic contributing the least to the stabilization. Okay, there is this question. So we have the anticodons. In which of these following? Anticodon. So codon is present in which one? So, and I had mentioned who is responsible for reading that part. So based on that, you can try to understand. Next is we have several statements. So could you pick the incorrect statement? And the most common secondary structure is which of these? And hemoglobin, this we have discussed. This is what kind of structure? So the first one is tRNA. So I'll just briefly show you again where is the anticodon actually present. Yeah. So as I had mentioned, this was the mRNA and this was the three codons. So anticodon is something that has to read this codon, right? So there was a tRNA that was reading it. So the tRNA was present. Something you will see tRNA of this structure here. So this tRNA will read this amino or codons based on this codon by the presence of anticodon, because anticodon is what will read these codons or the bases here. So the anticodons is present in the transfer RNA. Sucrose is a disaccharide which is formed from two glucose. Uracil is a pyrimidine, which is correct because it's a six-membered ring. Cellulose is a polysaccharide. You have several constituents of these glucose coming together. Alanine is, on the contrary, are not sulfur containing. Sulfur containing is, you can go back and check, if we have cysteine and methionine. And the most common secondary structure is alpha helix, and hemoglobin was a quaternary structure. We had discussed this. In the same session, we had also discussed about cellular metabolism. So cellular metabolism, I'll just briefly, not going into too much detail, just overall I'll tell you what does it mean. So we taken food, and we eat food so that we can gain energy from this. And why do we need energy? Because we need that energy to drive cells. So that is why it is called cellular metabolism, because the energy that is generated is used up in several activities of the cell. And you have these different processes which use up different raw materials. For example, this what we'll discuss is generally sugar, but you can also expand this to lipids, proteins. All of them will be metabolized in such a way that they can generate energy in the form of ATP that is used by the cells to carry out further activities. So I'll just discuss how glucose is finally translated to energy, and why this happens via two like spanning over two important pathways. But that we had also done this for lipids and proteins. So glycolysis, we had discussed this in the previous session. Glycolysis means breaking of glucose. Very easily to understand, it is a breaking or the breakdown of glucose to yield energy. So you start with glucose, because I said it's spread over two pathways. The first pathway, which is glycolysis, is ends up into in the formation of pyruvate in this intermediate step here, pyruvate. So you can see that you start with one glucose, you get two pyruvates. And the most important factor here is that each of these are maintained by or carried out by very specific enzymes. And all of these enzymes are regulated. What it means is, you can see here. So for example, here, transfer of glucose, or the first reaction is glucose to conversion to glucose, or conversion to glucose 6 phosphate. For this to happen, the body has to have enough glucose so that the glucose 6 phosphate, the ultimate step here is the breakdown of glucose. So your body has to have that many glucose so they can break it down. So high number of glucose will propel this pathway further. So extra glucose, and then you have ATP. You need ATP so that you carry out the first synthesis. This process is to generate ATP. What it did to happen, also you need ATP. So you need more of ATP. So more ATP, more glucose, you have this reaction in the forward reaction. And then you see this several reactions which are in the opposite reactions as well. So this goes in the forward reaction, this also goes in the backward reaction. These are the steps which are regulated, or which are called under being under regulation, means they're under feedback control. We had read about feedback control, which meant that a certain concentration propels the reaction in a certain direction. More of glucose 6 phosphate will propel this in the forward direction. More of fructose 6 phosphate will propel in the opposite direction. Why could it happen in the opposite direction? What will happen in the opposite direction? You have more of glucose formation. When will more of glucose be formed? When you want to store glucose, or when you want more glucose, which because you are not taking glucose in from external sources, so the body is trying to make glucose. So these steps will go in the reverse direction if you have to store glucose or you have to make new glucose. But if you want to break glucose, you have to go in the forward direction. So concentration of these ATP, these substrates, NADH, these regulate the several steps. So the ultimately for glycolysis, you have one glucose giving rise to two pyruvate, two ADP, two NADH, and yeah, four ATP. ADP means, so ATP is triphosphate, ADP is diphosphate. So you have four ATP, but two ADP and two NADH. Now, following it from there, now one pyruvate gives rise to one acetyl coenzyme A. This acetyl coenzyme A will be further used up for actual generation of ATP. Here, this transversion of pyruvate to acetyl coenzyme is again propelled by these kind of factors, whose concentration regulate the direction of the flow. Here, when it is converting to acetyl coenzyme, it will give out carbon dioxide, which is again given out. Then this acetyl coenzyme A is taken into something called tricarboxylic acid cycle or Krebs cycle, which ultimately will give rise to these NADH, FADH, and ATP. So if you see, you ultimately have 6 NADH, 2 FADH, 2 ATP coming from 2 acetyl coenzyme A, and 2 acetyl coenzyme A coming from 2 pyruvates. So if you see, uh, so one NADH gives rise to 3 ATP, one FADH gives rise to 2 ATP. So these are the total ATP that is generated from one single glucose, and you need that much of energy to drive a cell in cell activities. Okay, so very briefly, I have discussed. We'll try to understand this concept more by these questions, but you can always go back to the original PPT. The first step is mediated by which of these enzyme. Second is very simple. It says glycolysis refers to which of these process. Third says the activity of the enzyme increase when the ATP of the cell depletes. So you might not, if you have just looked into the pathway, you might not be able to guess it. But when I come to this answer, I'll go back to the pathway and show you the answer. And glycolysis takes place in which of the cell compartment? Yep, exactly. So the first step is hexokinase, which was the conversion of glucose to glucose 6 phosphate. Second, as we have discussed, glucose to pyruvate is called glycolysis. Third is phosphofructokinase. So just for the people who could not answer, this is where phosphofructokinase is, and you can see that ATP is affecting the activity of this enzyme. So that's why the question was, if ATP is decreasing, which of these enzymes activity will increase. And then you have glycolysis, which was generally is it is so in nucleus you don't have this, you have it in cytosol. Golgi and lysosome have their own functional activity. Okay, so we have these further questions. Okay, single molecule of glucose gives how many acetyl coenzyme A, which gives rise to a Krebs cycle? And the acetyl coenzyme A is formed from pyruvate by what reaction? So in the reaction which is from the pyruvate to acetyl coenzyme A, what kind of reaction that is? Next is in the given components. This is one of the intermediates of Krebs cycle. All of these, but they are also used for the formation of amino acids. So which of this could be such of the intermediates? And the entry of pyruvate into the TCA cycle is inhibited by the presence of high cellular concentration of which of these factors? So the first is correct. So you have two, one glucose giving rise to two pyruvate, one pyruvate to one acetyl coenzyme A. So two pyruvate to two acetyl coenzyme A. Acetyl coenzyme A formation is by oxidative decarboxylation. I'll go back to that slide just to show. So here you have decarboxylation under the presence of oxygen. That's why it's oxidative decarboxylation. Next is this alpha glutaric acid, which is further recycled into the amino acid process. And then you have the entry of pyruvate into TCA is inhibited by high concentration of what? So here you can see that NADH is what is propelling this formation. So if you have more of NADH, this is not this is inhibited because you need more of NAD plus so that this reaction takes place. Okay, and so this is the last segment of today's discussion, which is on central dogma of life, which starts with the basic description of how the three molecules are connected: DNA to RNA and proteins. DNA is transcribed to RNA and RNA is translated to protein. DNA is self-replicate or replicated into its own copy. And but just to mention here, when you go to the original PPT, you will have several of organisms on which there is a deviation from central dogma. So you necessarily don't have to have a DNA to form RNA. You can form an RNA from an RNA. You don't have to necessarily have DNA to RNA to protein. You can directly have DNA to proteins. So all these were discussed on those slides. Okay, so moving from the first topic, which is DNA replication, in very, very brief, DNA replication has very requisite machinery, which The idea of DNA replication, as I have mentioned, that it is to replicate the DNA in its entirety. So how does that happen? That happens by the complementary strand synthesis. So you have these parental strand. Each of these strands are synthesized by an enzyme called DNA polymerase, which sorts to move from one nucleotide to the other nucleotide by the basis of complementary. So let's say for one strand, let's say you have this as the parental strand. So you, and the synthesis is always in the 5 prime to 3 prime direction. So it starts synthesizing from here. First base it incorporates on the basis of complementary to the first nucleotide. So it's A, so it's T here, and then it's G, so it will incorporate C, and this way it will propel further. The DNA replication is mainly carried by three steps: initiation, elongation, and termination. Initiation is done at the sites called origin of replication. As you can imagine, the name itself says it's the origin of replication. So there will be regions here which can melt away easily to give rise to this. So you, for DNA polymerase, you need place to come and sit. For that to happen, you need to have region between these double-stranded DNA, and these happen at these regions, which are called origin of replication, which have bases which are less bound, or which have less hydrogen bond, which happens in the case of A to T, because A to T are have two hydrogen bonds and G to C have three hydrogen bonds. So these regions, these origin of replication regions, have more of AT reach regions because of which they melt away easily, and when they melt away they give rise to these kind of bubble, and this bubble can now extend further when the DNA polymerase comes and sits at this junction. So that happens. Now the first thing is, for initiation this has happened. For elongation, first you have the primase to come and sit, and elongation is basically addition of DNTP, but DNA polymerase cannot add new DNTP on its own. It it will add only at after a certain number of nucleotides have been added. That is because it doesn't have something called de novo initiation or de novo elongation. So what happens is, you have some enzyme, something called primase, which comes and sits at the first base, adds few of the bases, gives rise to something called primer. Primer being a short nucleotide stretch which is synthesized. Once you have this short nucleotide stretch, which is called primer, then the DNA polymerase can come and sit at this junction, and it can elongate by adding the enzyme, by adding the nucleotides. This is because when you have a primer template junction, there is a 3 prime OH free, and it can DNA polymerase then use this 3 prime OH to add bases. How does the base, so the direction is 5 prime to 3 prime. So if there is a 3 prime OH, it can remove, it can add base at the 3 prime OH, and again at by, so what I mean is, so let's say it's here, so it's a 5 prime end of this nucleotide, it will be added here, and then 3 prime end will be again free. So phosphodiester bond, which is the backbone of the amino acid DNA here, this will always have 3 prime at this prime, and at this end, and 5 prime at this end. And because DNA polymerase cannot use this property de novo, that's why you have another enzyme that starts the process, and then elongation happens when the DNA polymerase takes over. And the other factor is that in two strands, the elongation is not equal. So in one of the strands, let's say for this strand, the elongation can take place easily in this direction, 5 prime to 3 prime. Why is that? Because a template strand has to be anti-parallel. So the template strand is of this nature, and 5 prime being on this side. So the strand is unwounding at the behind, 5 prime end is opening, and this strand is moving continuously towards the end. But on the other strand, because DNA inherently is anti-parallel in nature, this would be 3 prime here, and this would be 5 prime here. So the synthesis will always happen in this manner, like this, like this. So this are called fragments because each time this unwounds, there is a new 3' end. So it will sit here. So what I mean is the polymerase will sit here and it will start synthesizing this side. Again, a certain region will open, it will sit there and it will start synthesizing. So these are little fragments. These are called Okazaki fragments, and that's why it's called a lagging strand, because it is not continuous as opposed to the leading strand, which is a continuous synthesis. This is an important feature. And at the end, you have termination sites. These are different for the prokaryotes and eukaryotes. Basically, these are the sites where the DNA polymerase will move away from the DNA strand, and this could be because of presence of certain sequences or certain sites. So let's say here you have these termination sites, which is in prokaryotes. So that when the DNA polymerase reaches these sites, it gets an information to leave the DNA sequence, and as it moves away, you have the new strand synthesis completed. But this is slightly different in the prokaryotes, which will use a different mechanism for the DNA polymerase to leave the DNA and the so the new and the old template junction. So this is the difference. Like there are different DNA polymerase which are present in eukaryotic and prokaryotic system doing different function. So this DNA polymerase 3 is what is used for replication in the prokaryotic system, but DNA doesn't always. So in the if it has to do polymerase activity, it will do 5' to 3' activity. But the DNA polymerase also has something called exonuclease activities, which is removing of nucleotides in the opposite direction. This is important for proofreading, which is an inherent checkpoint of a cell. But in prokaryotic system, instead of being 3, there are 5 polymerases carrying out different activities. So one of them will carry out repair, then one of them will carry out a proofreading activity. Mainly the replication takes place via this, but you can see that for two different fragments, two different strands, you have two different polymerases working. And the idea of the DNA polymerase reaction is used in the PCR, where you have this same DNA polymerase reaction taking place at a faster rate over several cycles, so that your template DNA, which you start with a lesser number, by the end of your cycle, let's say generally it is put for 35 cycles, you have multiple copies of your strand. And this is possible because of the use of primers, which are very specific to a region that you want to amplify. So let's say in this whole template you want to amplify in this region, so you give something called primer, which does the initial which goes and binds to the regions, gives that initial primer DNA junction, and then you have DNA polymerase replicating over only this stretch continuously for, let's say, 35 cycles, which exponentially increases the number of these wanted genes, and we have an end product with high number of concentration of the wanted gene. So this is same steps are carried out in PCR, where you have elongation so elongation and termination, but the initial step is denaturation and annealing. So annealing being the primer attachment to the DNA, and denaturation being the opening up of the DNA. So let's say you start with this DNA, you open and make it to a single strand, anneal the primer, extend it, make it something like this. For this new strand to undergo amplification, it again has to form two separate strands. That is again done by denaturation. So that's why it keeps on going in cycling, and that's why DNA or PCR is also called thermocycler or thermocycling step. Okay, moving on to transcription. Again, transcription here means that it is the formation of the mRNA from the DNA, and it happens when there is a gene expression to be taking place. So here you have a transcriptional unit, which is a DNA sequence which will be transcribed into RNA, and this RNA here is called the messenger RNA. This will have several information that when translated gives rise to the protein, which is important. Now in eukaryotes, you have monocistronic transcription unit. Basically, you have one promoter or only one unit which is giving rise to one mRNA. But in prokaryotic, you have these several coding sequences lining up together. So you have several like cistrons in the region which is transcribed. So all of them are together only. So in one contiguous thread, you have several amino acids or proteins being translated or transcribed for. Again, transcription also carries the same three steps, which is initiation, elongation, and termination. Initiation being the RNA polymerase coming and sitting on top of it at the site, which is called promoter. Promoter will allow help the uh, so it's promoter is at the 5' end of this gene. Okay, so here the polymerase will come and sit. There will be a start point. So start point is basically where it can start transcribing from. So this will also take me in the 5' to 3' direction, as you can see here, and it will start transcribing until it reaches the end point. The end point, which we'll see, is based on some structural changes to the mRNA. So it will first start, this is called initiation. Again, you can see for it to initiate, it has to enter, it has to be part of the double helix, so it has to be open up. So in this, when the mRNA is moving ahead, the DNA will start forming helix again at this rear end. So mRNA is moving behind, DNA is getting rewound at the back. And as it move ahead, this bubble keeps moving ahead and the transcription takes place. And so this is elongation until it reaches the end. It continues, and at the end point, it has termination, which again is based on several cues. And the cues are these post-translation modification. So when a mRNA is transcribed, it is called heteronuclear RNA or heteronuclear hnRNA. On this RNA, you have several processing. You have a poly A tail, so you add a poly A tail to it. You have a cap, you add something on the 5' end. So the 5' end and the 3' end are like, you can say there is changes modified. And then you have introns. So in DNA, eukaryotic DNA, you have several regions which are not transcribed, which do not code for anything. So for a particular mRNA to code for a meaningful protein, these these regions which are interspersed between the exons, these needs to be removed. These are called introns. So you have this splicing of intron with the the procedure that happens is called splicing, and the machinery that it takes place or that carries out splicing is called spliceosome. So these introns are removed. All these the exons are what codes for the amino acids or the protein. So these exons are all stitched together, and you have poly A signal and 5' capping. Poly A tail, addition of poly A tail says that okay, this mRNA is completed now. You can remove this, and this poly A tail is at the, imagine you have poly A. When can a poly A be added to the amino acid or the mRNA? When you have several of these TTTTT in the uh, DNA. So in place of T, you add several. In place of several T, you add A. So you have this poly A chain on the mRNA, and that gives a transcription termination site. So you can see right before the termination, you have the poly A signal. So that is the cue for termination. So these are what gives the termination signal to the mRNA or transcription, which is then taken to translation. As I have mentioned already, the translation is where you transcribe this mRNA to protein. For this, what you need to remember is it reads like this. So it has three reading frame. Three reading frame means 1, 2, 3. So it can start reading in triplets at three different positions. It can start reading from AUG. It can start reading it as UGA or GAC. Why not fourth? Because if it starts reading from ACE, it is as similar as the first one. So there are three reading frames. This is from the front. So from the from the first side, and you can also read it from behind. So that would be from the reverse side. So there are in total there are 6 reading frames from plus direction and from the reverse direction, forward direction and reverse direction. And you can see here each amino acids, it's coded by one codon. So AUG, as you can see, will always code for methionine. But there is something called degeneracy. So one codon can code for only one amino acid, but several codons can code for one amino acids, like single amino acid. So what I mean is UUU will always code for phenylalanine, but phenylalanine can be coded by two codons because there is something called degeneracy of codons. So these codons are redundant, so they can code for one amino acids. But to remember is that one codon will always, or one codon will always code for one amino acid no matter what. It cannot code for two amino acids, but that's why you have 20 amino acid but 64 triplets, 64 codons. So there has to be redundancy, among which these UAA, UAG, UGA are called the stop codons. So translation stops at these signals when you reach, when the mRNA when the mRNA is reached at these stop codons, translation stops. And as I was mentioning in the previous slides, that transfer RNA has the anticodons. Our RNA makes up this ribosome. So ribosome, mRNA, and then ribosome has these three different sites: E site, P site, A site, which are responsible for protein synthesis. So you have these amino acids added from this site, transferred to this site to the growing chain, and this amino acid is added by this transfer RNA based on what kind of code it is reading here. So all these transfer RNA has these kind of codons here. They will try to match this codon which is on the mRNA. Once it matches, then obviously it is bound to the respective amino acid. Once this amino acid fits into this cleft, it is transferred to this site. At this site, let's say it's methionine, valine, lysine, it will now add valine to it. It will keep growing until it reaches a stop codon here, because of which there will be no amino acids added by the transfer RNA, because there is no amino acid for those codons. So this will receive a cue for stopping or terminating, and the translation will thus be terminated, and the peptide chain which was formed here will be released. So that's exactly what is happening. It is little bit more complicated where you have these two units coming together and forming. So this is all described in detail, the respective PPT. Okay. To summarize all of these transcription, translation, and replication, I have compiled these set of questions. So we'll try to do that. The first thing is, again, the nucleic acid synthesis takes place in which direction? The eukaryotic replication of DNA is of what nature? It is easy if you just do it like if you remember the diagram we discussed. The primary control of gene expression takes place at the level of which of these steps? And the anticodon of the initiation codon. So AUG is the initiation codon. AUG, methionine, is always the first amino acid to be translated. So the anticodon for this codon, initiation codon, will be what? Yeah. So the first is 5' to 3' direction. Second is bidirectional, as you have correctly guessed, that it would open as a bubble and it will move in both sides. Third is transcription. Fourth is anticodon. So, okay, let's try to do this. It should be easy. If it is AUG on the codon, it has to be anticodon. So, it has to match itself. So, it has to be A, U, A, and C, because this has to bind to these codons on the mRNA. So it will be UAC on the transfer RNA, which will bind to AUG, and then it will bring the methionine onto the growing polypeptide. Okay. And the last set of question for today is, in eukaryotic, RNA2 facilitates the transcription of what? Nonsense codons. So these codons means basically sense codons are the ones which are translated. So nonsense codons will be what? And in a transcription unit, the promoter is located where? And spliceosome, you generally don't find spliceosome in which of these organisms? So the RNA2 is responsible for transcription of the HNRNA. HNRNA means the heteronuclear RNA. Nonsense codons are the codons that do not code for amino acids. So as it says, that it controls the termination message. Third is a transcription unit has the promoter at the 5 prime end. We did this. And spliceosomes are not present in bacteria. They are present in all other advanced organisms. And just to go about this, so if you have RNAs too, which are present in the eukaryotes, then you have them translating or transcribing different products. So RNA polymerase 1 does for rRNA, RNA polymerase 2 does for nuclear pre-mRNAs, 3 is for transfer RNA, 5 is ribosomal RNA, 4 is for small interfering RNA, and polymerase 5 is for in plants. So the 4 and 5 you find in plants, and they are for the small interfering RNA. So as the question says, RNA is 2. Nuclear pre-mRNAs are also called heteronuclear RNA because they are not cleaved. They are still with the introns and the exons. So that's why they are called heteronuclear RNA. Okay, so that wraps up today's summary session. The remaining of the topics I will do in the next session, which will be in the next Tuesday, and then we'll cover the remaining problem solving sessions with where we have covered so far. Thank you.